Welcome everyone, I'm Daniele Perlo. I'm a PhD student from University of Turin, and I'm currently working in the computer vision lab called ADOS Lab in the computer science department. Today, we are talking about dysplasia grading of colorectal polyps through convolutional neural network analysis of whole slide images. In this presentation, I would like to show you why focusing on dysplasia, the classification is so important. And after that, I show you the data set we are collecting and the method to make the prediction, the classification, and some other findings. At the end, I show you some results, some inference performance on whole slides, and some visualization to you to better understand our work. Histopathological diagnosis of colorectal polyps is a key factor to early diagnosis carcinoma in colorectum. Whole slide scans are high-resolution images of biopsies, and that can be used for automatic classification tasks. Data is collected within the European project DeepHealth from the Unit of Pathological Anatomy Department in Turin. The dysplasia is defined as the abnormal cellular development of an organ or a tissue, generally consisting in a loss of control mechanism with the replacement of metal cells by emitter cells. It can be a condition that predisposes the neoplastic process. Why focusing on dysplasia classification is so important? Because different chromoscopic surveillance routines have to be set to prevent carcinoma in patients. And the dysplasia grade is a major risk factor which highly influences the routine choice. The glandular dysplasia can be divided in 3 different grades: low, medium, and high. Middle dysplasia grade, for example, is not reported in each medical institution guidelines due to the difficulties in concordance between the pathologist itself. In this work, we take in consideration different type of tissue. Besides the normal tissue, we have the hypoplastic polyp and tubular and tubular adenomas with high grade and low grade dysplasia. Working on whole slides means working with high resolution images in which there are the projection of tissue taken from the biopsies. We ask to all pathology not only to label with the entire slide with diagnosis, but also to highlight, to annotate what we call region of interest. So informative tissue that help them to make the diagnosis. We know that working with neural network is infeasible to feed them with high resolution images. So we develop our pipeline to working into patches, small crops of the relevant tissue, and we train on that. Talking about our data set, we can see how the number of samples for each pathology is related to the pathology itself, to the frequency of the pathology itself. And also the tissue, the relevant tissue that we have in each single slide is related to the pathology itself. This data set also includes the whole slide sources of the public available Unito-Path data set. We divided our data set at the slide level because one slide means one patient. So we have no overlap between train and test patients. We also have a validation set, but is make at region of interest level. And the region of interest are taken from the training set. Now I will show you our work pipeline. An image for the training is a region of interest, that is slicing we slicing them into patches, pre-process those patches, and feed the neural network and train on that. And we have a prediction for each single patch. At the inference level, we have the tissue in the whole slide, and we slice them also into patches, pre-processing those patches, feed the neural network, and make the prediction for all the patches. After that, we try to predict the whole slide diagnosis by maximum voting criteria from the patches prediction. Our model is a ResNet that was ImageNet pre-trained. We train it for 250 epochs, and we choose the best model in terms of balance accuracy on the validation set. We use also a lot of data augmentation that we include vertical and horizontal flips and some random operation like rotation, equalization, solarization, inversion, and contrast enhancing. What we find that is that we have to work with grayscale images. Let me explain this graph. This is an excursion in physical dimension of the patches. And we find that working with the grayscale patches images does not remove useful information, and on the contrary, helps in removing the color biases. That is the blue line, and the RGB is row 1, is the orange one. The same goal is not reached by using tissue normalization techniques, the green line, like Machenko. As we expected, also in inference for the whole slide, we have the same boost of performances if we use grayscale images. For example, we can see the hypoplastic polyp and the normal tissue that have better accuracy for the same physical size of the patches. Now I would like to show you some metrics, F1 score and balance accuracy. And these graphs, each line is a different class, so we can see the score for each class separately. We can see how for some classes like normal tissue, if we enlarge the patches, the physical resolution of the patches, we have more high accuracy. But instead, by enlarging the physical resolution of the patches, we decrease in performance for other classes like high-grade dysplasia or hypoplastic polyp. So there is not a correct solution for each type of tissue. Now I would like to show you some visualization. For example, there are 4 different images for different classes. The first one in white is a normal tissue, is mostly white. The second is hypoplastic and is mostly red. The third one is a low-grade dysplasia, is mostly green. But the more interesting one is the fourth one, the high-grade dysplasia tissue, that also show a huge amount of low-grade dysplasia feature. We also validate our model by to our pathologists by showing them the attention map of the neural network that is taken with the GradCam algorithm on the patches. And we can see, for example, for hypoplastic polyps, that is the top right corner, and that the network is focusing on the serrated gland. For dysplasia, in the bottom right corner, we have an example of high, high grade of dysplasia, and we can see that in the network is focusing on the paraneoplastic lesion. Thank you for your attention.